I'm going to ask some questions, and I have questions for all of you, but before we go to the questions, and we have some that you all have called in as well, but I would love, just very quickly, if I could, and we'll start here with you, Catherine, and make our way through. I'd love your thoughts, because conferences like this are an opportunity to hear from parents, to hear from one another. What are your thoughts? What will you take back with you from today?
I think it's been a wonderful day. It's wonderful to see so many enthusiastic, motivated people in the room, and to feel like there's so many people working towards the same aim, and it's such an important aim. I think for me, the most fantastic thing about the day was to hear the various parents speak about their experiences, about things that have been so tough, so life-changing. I think Danny had half the room in tears this morning, as did the rest of you, and you put so eloquently how life-changing these conditions are, and it's really important for people like me to hear and to be reminded about why we do the work we do. So thank you.
That's wonderful. Thank you. Anne?
Well, as someone not from Australia, I think I'll just continue to reflect on how impressed I am by the community, and by that I mean the partnering community between the families, the researchers, the physicians, and by how cohesive it is. So I think what I'm taking back from my whole Australian sojourn, but particularly Geta as an example, is how to do this, and I'm trying to take back that spirit of collaboration. And we're one team, whether it's many genes in different parts of the community. We need to be one team if we're going to make progress. I think that the biggest thing I'll take back is how much work we really have to do, but we really only can do it as a team.
I think one of the things I'm constantly marvel at is the amazing parents. And I just feel so privileged to work with such incredible families. I reflect on the fact that we've come a hell of a way. In 2001, nobody believed these disorders were genetic. And in fact, when I went to do my PhD, when I left Great Ormond Street in London, they laughed when I said I was going to do it in genetics of epilepsy. So boy, have we come a long way. But the other thing I really think is we're still at the tip of the iceberg, and the gene is just the beginning of an answer, and we've got such a long way to go. And I'm so thrilled to have these very talented people that I work with, and I can see them, they're going to change the world, and I think it's really exciting.
Similar thoughts with what Anne said, coming from the United States, arguably considered one of the most powerful countries in the world, and with tremendous amount of funding for science and lots of people passionate about many things. I'm awestruck at what you have done in your country and what the potential is for you to move the world forward. And as I said to the young people yesterday who are engaged in research, think big. Don't think small. Think big and just reach as far as you can possibly go.
I guess for me it's sort of a similar theme in that it's energising for me as an Australian scientist, because we are out of the loop. And and as Ingrid will tell you, we get on the aeroplane. I've got 1.3 million miles on my Qantas account.
Can you give some of them to me to go see my daughter in America?
Let me tell you, they make them very hard to spend.
No. I know deserving people. Yeah. Well, I'll work with you on that.
So it's a lot of energy. It also energises us to think that we can do anything. We because we are, it's a very small community of epilepsy researchers in Australia relative to the community in America, and so you feel as though, well, you've got to sort of take on more, and so that energy is important for us to think that we can take on 6 or 7 big projects in the lab to try and help as many people as we can. That energises my lab. A lot of them were here today, so I think that's something that's really important for me. We don't see patients that much, and so it's much more acute and poignant for us to feel this and to feel the energy and the stories like Danny's and Danielle's. I see them socially, but I don't see that side of it very much, and that's amazing for me to see that.
Emma?
I think one talk I once heard was that we get nowhere if we think that we have to start from the beginning each time. And Ingrid put up that slide very early on about let's not reinvent the wheel. There is so much immense knowledge from you, from your families, from the experts here, and we can just go so, it's unstoppable if we work together and we learn from what we've come from. So thank you everyone for being today.
So I can't tell you how much that means to me because it is a love fest, but I will push you. I do love a love fest. I really do. So now let me be mean. So why aren't we further, and I don't mean this in a mean way, I genuinely don't, but we have the smartest people in this field right here, right now. So just quickly, again, round robin, what's holding us back? What are we doing wrong that we need to fix? Emma, I'm starting with you. What are we getting wrong?
So I think we can work more together.
So what does that specifically look like?
Specifically, I think you're hearing today how a lot of families said, I wish I knew that my doctor was giving me the right advice for my child with this particular condition right at the start. So reflecting on what Ingrid and Catherine said, why can't we work together as a team and get the least basic standard of care out there and work together, as Dan said this morning, to educate the medical workforce and bring that up. So at least we're starting.
I mean, the easy and obvious answers are more money, but that's not, I mean, that's just a part of the problem. I think one of the barriers, and things that are slowly being addressed, it's the lead time. I mean, Dan and others started EPGAP that fed into Epi 4K and to others years and years and years ago. So it's to ensure that we've got this pipeline of things that started, that are highly collaborative, highly networked, that started 10 or 15 years ago, and for us to be thinking today, what are we doing for the next generation?
Well, and
making sure we're laying the foundations.
I want to come to you, Dan, just one sec before Steve hands over the mic, because, you know, Silicon Valley, the whole thing is move fast and break things. Are we just moving too slowly and not breaking enough stuff? Either of you, Dan, you can go with that. What do we need to break, or do we need to create more?
There's no one answer to this question, and I believe there are a number of touch points, and we'll probably hit on each a unique one. I'm going to throw in the very strong belief that our future is grounded in our ability to understand this incredibly complex entity called the human brain. I'm a real believer in the power of basic science, and from a historical perspective, the leaps that we've made in terms of effective therapies have all essentially been grounded in advances in our understanding of basic science. So I think that's one required facet, not the only.
So as we try to do the kind of shiny things, the exciting things, the sexy things, we can't forget the kind of undergirding. I see nodding. Ingrid, is that true?
Yeah, I think that the basic science is what informs as we move forward, and is what's really makes a difference, which is why it's wonderful to have physiologists like Steve really explaining it so we can understand it, and really trying to develop the right targeted therapies. My view about what we're doing wrong, and we've heard it today loud and clear, is we're not working as an Australian-wide team. I feel very much like we have been very fortunate to have people collaborate from all over the country over the last 25 years, but I can hear now that's not working very well. And one of the things that went wrong with my slides and computer decided to stop was that in this plan for the natural history study, we want to have an Australia-wide network, and we're going to have a neurologist in each of the capital cities that's sort of the leader for that area.
Including Adelaide? We hear you, Adelaide. We hear you.
So
that has come through.
That's on the agenda, but we need people to make this happen, to get the work done. But the idea, I'm hoping, the Australian Genomics that Emma mentioned, the epileptic encephalopathy flagship, was the beginning of that, but that funding finished at the end of last year, and now we're just looking at the results and seeing how many children were solved with that. It's about a 30% hit rate, I think, something like that. But really moving forward, if we can engage the country more, hopefully we can upskill everyone. But the problem is we have to reach all of the neurologists and all of the paediatricians. We had a paediatrician from Adelaide who came today because she's got a bunch of patients who are relevant to this community. But most of the paediatricians, they're not here. So I think it's really important that we engage our colleagues, and we do a lot of education. We all do a lot of education, but we have to do a lot more.
All right, Anne, what are we getting wrong? What are we doing wrong that we needs improvement?
Well, I'll start by saying I absolutely agree with everybody and all you've said, and particularly the need to ground ourselves with more basic understanding. But I think what, at least in the US, we've done wrong, but I guess it probably applies to most places, perhaps other than Melbourne, is that the crosstalk between the clinic and the lab has not been deep enough and strong enough, and it's been sort of portrayed as these separate entities. You've got this clinic on one side of the street and this lab on the other side of the street, and it's often different people who don't get together on a regular basis. So I think the culture that we're trying to create, where we have some of the same people both in the clinic and the lab, and a culture of training people who are actually clinician scientists, where some of the science that they do is clinical science, the science of natural history, the science of clinical trials, talking weekly, daily to the people who are doing the bench science. I think that'll move us forward. We certainly have not done that actively enough, either in our training or in our practice in the US.
All right, good to hear these things. Catherine?
I think, so Dan, I think touched on the issue of lead time to things, things taking a lot longer than you think, and I think one of the things that maybe not doing wrong right now, we can see that we will be doing wrong if we don't plan for it right now, is trying to work out at the point of which we start to have other treatments available, lots of clinical trials and things coming through, what kind of workforce do we need to make sure that that we're prepared for all of that? We're going to need to start training people now to make sure that we're set up for that kind of thing, and so it includes neurologists, genetic counsellors, basic scientists, other medical specialists, therapists, etc. All of the people that might be involved in clinical trials and then eventual translation to routine clinical practice. So it's something we need to work on now.
All this is very interesting. I'm going to actually do a bit of a jump cut, and because I'm going to move around between you guys, but I want to go to you, Dan, because when we talk about shiny objects, and one that was shiny and we didn't get to... I didn't do that. Self-inflicted. That was so self-inflicted and very, very funny. I told you, he's like the complete package. He's super smart and super funny, in addition to all that exploring. So here's my question for you. When we talk about exploring, who knew how interesting the gut was? Yeah. Okay? So the microbiome, you were talking a little bit about that. You promised us more. This is your opportunity.
Okay, so yeah.
Expound.
It's, as I said, it's just a remarkable discovery that we didn't know up until about 10 years ago, and that is that every single one of us has this unique extra organ that's made up of 3 trillion bacteria, and every one of ours is different. And it's only been recently that we're beginning to appreciate how much the variation in our microbiome from day to day, week to week, month to month may have an effect on all of the other organs in our body, including the brain, which I emphasized with one of those slides. But here's what sort of blows my mind about this. Everybody knows we have an immune system, right? And there are these cells that travel around, and they're looking out for foreign invaders. They're called B cells and T cells, and they produce these chemicals that circulate around in our blood, some of which get into our brain. The immune system is arguably second only to the brain in terms of how complex it is. It's actually sort of like a liquid network instead of the more solid network that's up in our skull. But it's incredibly complicated, and it's constantly doing this surveillance. But here's what I didn't realize until just about a year or so ago, and that is the foundation of our immune system is set up the moment of birth.
Okay, yeah, is everybody taking that in?
Okay.
The moment you're born?
So when you're a fetus in your mom, you're basically protected from the outside world by your mom. So you don't actually have an activated immune system. I mean, the cells are there, but they're just waiting to be given the information as to exactly what they should respond to, right? And it's only when you pass through the birth canal, or if you have a C-section, you get exposed to the outside world. In that instant, that immune system is seeing the outside world for the first time, And that sets the baseline for the rest of your life. And so here's the experiment that I was mentioning to Sarah. So there's an investigator at UCSF who asked the question, if we look at infants and look at their microbiome, and just to make sure, in order to assay, to understand what your microbiome is, you have to have a sample of poop, right? And so you analyze the poop. So she took a group of about 200 infants who were born and sampled their poop at 2 or 3 months of age and then followed them for a number of years. And the hypothesis was, are there any immune-mediated diseases that are going to occur later in childhood that might be related to the makeup of the microbiome at 2 or 3 months? And she was able to show with 80% accuracy which children were going to develop asthma at 2 months of age, when the asthma actually didn't appear till 3 or 4 years.
So so this is so fascinating. You see why I wanted to come to this. So what are the applications and implications of this for epilepsy?
Right, so we are only beginning to know, because there's essentially been no research done on the microbiome and epilepsy. I checked this about 6 months ago. If you do genetics and epilepsy or seizures in PubMed, 6 months ago, I think there were 23,000 publications. That's no surprise to any of us. If you do microbiome and autism, 200 publications, because people have recognized that the immune system and inflammation may be associated with autism. We know that inflammation is important in epilepsy, but again, 6 months ago, it's probably a few more now, there were 17 publications total of epilepsy or seizures and microbiome. That's it. One of them is this paper in Cell from last June.
And just because this is a very savvy audience, we're talking about Elaine Chau's.
Yeah,
so this is from UCLA.
Yeah.
And this, the scientist is Elaine Chau, and that's
HSAIO.
HSAIO, for everybody who's out there listening. So you're going to want to have a read of that.
Yeah, you look her up, and it was a paper in Cell, which is one of the most preeminent journals in science.
And then the other question that I want to ask quickly, and then we'll move, because other people may want to weigh in on this, but the other thing that instantly occurs to me is, I'm going to show of hands, here's mine up, how many of you who have children with genetic epilepsy have some sort of issue with what's going on in the gut?
Oh, yeah. Yeah.
All right. So, in other words, is it your suspicion that you may find that there is something important that we can learn from it?
There's no question about that. It's just that we're right at the beginning. And Elaine's study showed that you can take 2 specific bacteria that exist in the world and are in people's guts. And those 2 bacteria, if you implant them into a mouse, are the equivalent of the ketogenic diet. So in the future, I think we're going to be manipulating our microbiome in a way that will allow for us to enable certain therapies that we currently are so difficult, for example, to be able to offer. Yeah.
Such as?
Well, something equivalent to the ketogenic diet. But then also, how many times do we take a pill and the ability for the pill to have its effect is being altered by the gut or the way the immune system responds?
All right, so I'm instantly thinking that possibly Anne, Ingrid, or Steve might want to weigh in or have a thought on this. Any of you guys? Are you up on this, or shall I come to you with the other?
So just make a quick comment just in terms of sort of, you hear these new things, and it's a pretty much of a curveball, I think, and it took some time for this to sink in. But I think in terms of other examples of the important effect of the gut on the nervous system, and how can something down here affect this protected space that we're saying is so hard to get to. There's parallel studies in the Parkinson's disease literature where it turns out that if you've had your appendix out, you have a much lower risk of having Parkinson's, which I found out when I had my appendix out, and my husband was like, "Good news!"
Can we protectively do that? No.
But personal stories aside, I think it does tell you. We sort of think of the appendix as this vestigial organ, but we know that there's probably an immune role, and there's an involvement of lymph cells and so on. And so, but who would have thought that something that happens usually when people are kids, it's sort of a bit of an afterthought, but you take it out, no big deal, could actually have a long-lasting effect on the development of a nervous system disease years later. And we don't know why, but I think it's just another powerful illustration of how there are unknown unknowns that we really don't know, and that this has been one that sheds light. And there's certainly precedent for the gut having an effect on the brain. And who figures that out is going to take another group of trainees and generations of people who really have at that specifically.
Ingrid, is there anything that parents out here in the audience should do right now, or not, based on, I mean, you hear these things. Is there any practical implications for this, or is this say, watch this space, which is a perfectly reasonable?
I don't think there's anything today you have to do to change your child's diet, but I do think it makes you think at a very simplistic level. If you change your microbiome, it might change how drugs are absorbed. That's sort of a no-brainer. And maybe if you have particular microbes there, your drugs get into a different level in the blood and therefore are more or less effective. But what's even more intriguing is we talked a lot today about a genetic mutation and a change in your DNA, and then there was a reference to, by you, I think, to higher level changes in the genome. So it might not be in a code, but it might be what we call epigenetics, how genes are expressed. And I think it's very likely that the microbiome could affect which genes are expressed, or perhaps how much of a gene is expressed. So maybe it is something that one day we could use to regulate these diseases.
Steve, do you have any thoughts on this? Or?
Sure, always. Let me just get comfortable.
Just lean back. We'll get you a glass of red wine. No, it's too early for that, Steve.
And I won't resort to Donald Rumsfeld moments. Unknown unknowns and known unknowns. Yeah. You look at it, I mean, the gut with the microbiome is a little chemistry set. And we're so careful when the new drugs are tested and all the issues around toxicity. We've got trillions of bacteria making all these chemicals. I don't even know what half of them are. And surely, and our body's very good at adapting to situations. So there are these things called homeostatic plasticity. So you can understand that why that interaction is going to be. We're going to be absorbing a lot of the metabolites that these bacteria are producing. And when you make a change with a diet or with any other intervention, that's going to affect what they produce, and that's got a massive ability to impact our system. So it seems obvious, like if you want to be an armchair quarterback and sort of say, okay, it's obvious now, but it's amazing that it took this long for us to really realise it. We still don't yet know. If we understood it, we could manipulate it and work it in our way. And that means we don't understand it because we really don't know how to manipulate it and turn it into an advantage, but we will.
Dan, you look like, did you have one more thing you wanted to say? And Emma and Catherine, I didn't mean to leave you out of this, but are there thoughts? Yep, okay, because there's other things I want to come back to. I know, it's a different thing. But while we're on this, one last question on this, because I do think it's important and it's new. I'm trying to remember because I read the article in Cell a while ago, but it seems to me that Elaine Schau said that the gut chemistry was changed in people who'd used antibiotics. Right. So can you, because again, this is territory that's familiar to everybody in this room. Our kids probably, seems to me, get sicker than most kids. We've certainly done our rounds of antibiotics. Any information on that from anybody in this?
I'm not a clinician, but I've had people in my lab who have been on antibiotics and then get an opportunistic infection with difficile. And so if you do take antibiotics and deplete the natural bacteria and you get an inappropriate one, you can get very, very sick doing that. So, I think the medical community knows.
Emma's got a comment on this.
I've got a comment, which is that I know how much I don't know. And I see a lot of families where they had the same genetic change and it affects one child completely differently from another child. And that's something we all know, but we would love to understand more. And surely all our parts of our body works in concert. And although I don't know anything about this, I do fermenting at home of vegetables and things. So I think maybe having a healthy gut's a good thing. I don't really know why, but I think it's a good idea.
Yeah, fair enough. No, all thoughts welcome on this. Anybody else who wants to weigh in on this topic before we move on to something else? So the next topic I want to go to is, because, I'm a journo, so we got to go ripped from the headlines. In the United States, just a couple of days ago, there was a big story about Bubble Boy. So these are, if you've seen the old movies, these are the children who have an immune problem whereby they need to be kept in a protective bubble. They can't see anybody or touch anybody because they're going to catch something and die. St. Jude's has just created through gene therapy, something that apparently it has turned this around for 8 patients. They are quote-unquote cured. Does anybody want to weigh in on this? Do you know anything about this? The gene therapy piece of this is very exciting. We touched on it earlier today, but there's room in this panel discussion to go into this in greater detail if anybody wants to jump in. Come on, water's fine.
Well, I mean, I don't know that specific story, but it's an example of where our understanding of the genetic basis of various cancers and immune deficiency diseases is now being translated into true gene therapy, because the easiest system, if you will, to go and manipulate in the human body is the white blood cell system, which is the basis of many different types of circulating cancers and immune deficiencies. So what's done is you can take a person, this has been going on for a long period of time, you can take out their bone marrow and generate revised white cells and then ablate the entire immune system in a person, which has been done for a long time. But now with gene therapy, we can manipulate the genes and put in essentially healthy cells, and
Well,
then they take root in the marrow, and they start dividing, and you basically replace your immune system.
And that's, okay, so you're basically right on the money. So it says, in the study published 17 April in the New England Journal of Medicine, researchers used a harmless virus to insert a missing gene called IL-2RG into bone marrow cells taken from infants with X-linked severe combined immunodeficiency disorder. And basically, they say they've safely used that therapy to treat 8 baby boys born with this disorder. So I guess let's go to the, obviously, our children have different issues, so that gene therapy aspect is exciting.
So this is the beginning, and I showed that slide from the Allen Institute, if you may remember. It was a picture of these different sections of the brain with different patterns of lit up cells. And the point there was they've been able to work out a means of using a viral vector and introducing a specific gene and putting it in the blood. I mean, it was injected into the bloodstream, but that virus found its way into the brain and then found its way specifically into subsets of neurons, distinct subsets of neurons. So there are a number of challenges to get from where we are with bone marrow transplantation to brain cell alteration, but this is the beginning. Yeah.
All right, anybody else want to weigh in on that?
Or just to say, and there are conditions like Sanfilippo syndrome, so that's a neurodegenerative condition where that type of approach is being used in Adelaide.
You're definitely a theme this year, Adelaide. You really are. I feel like we're going to have to get a song. You guys need to create a song for us or something. I have some questions that have come from our audience. Does a response, actually this comes up from earlier, we discussed this. Does a response to a particular anti-epileptic drug predict a genotype? So again, this is the kind of reverse engineering. Does anybody want to add a little bit more information on that? This was from the end of your talk, Steve.
I don't think one can make a straight diagnosis on the basis of a treatment, but I saw a patient in the last 2 weeks. I can't picture the patient, but I remember the patient, I think, was KCNQ2, and someone had used carbamazepine Tegretol, and it had worked like a charm, and it was complete chance. But in retrospect, we realised that that was fantastic because they'd used it, and the parents had recognised that. But the moment it's not specific enough that you could see something worked and then go back. And I think one of the things that is worth pointing out is it's much more complicated, these diagnoses, than just one thing. And I remember when I was first getting tuned into Dravet syndrome, which was around the time I met Danielle, which was about, I don't remember what year I met you, but it was about 1999 I diagnosed my first patient with Dravet syndrome. And at the time I was really getting excited about it. Charlotte Dravet actually described it in 78 in French and in 82 in English. So it took a while to permeate the paediatric neurology and adult neurology community. Anyway, when I was diagnosing it, my colleagues were beginning to get interested, and they knew that lamotrigine could make some children worse. I think I've got some children where it's definitely been good drugs, so I'm not a complete believer in that. But every time lamotrigine made a child's epilepsy worse, they said, it's Dravet syndrome. And the epilepsy might have started at 3 years, which is way too late for Dravet syndrome, etc. So it's just pointed out to me, really, that these are complex diagnoses and you can't go on just response to one drug.
All right. Any other thoughts on that? No? All right. So another question from our audience. What research is going on in DMN1? Yeah, this, by the way, did have your name on it, for Steve.
So there have been some studies looking at the mutation and the mechanism of that, and there's actually a dominant negative interaction that drives the disorder. There are ideas for therapy now, and I think it's one of those situations. DNM1 has some recurrent mutations. I think it's R46W mutation, so it opens up the possibility that you could make a mutation-specific drug in that situation. And I think that there's discussions and thoughts around that. So DNM1 could be something that has a therapy on the horizon. So a drug that will only act, so if you've got a mutant copy of the protein and a normal copy, the drug will only act against the mutant copy. And I think that would be the idea for a disorder like DNM1.
All right. And then speaking of specific disorders, this one is for you, Annapurna Paduri, from Tamer in Egypt. Are there drugs being developed for disorders like IQ-SEC2?
So everything seems to be coming back to Adelaide today. So I was just in Adelaide meeting with our colleague Cheryl Showbridge about and hearing about her project with IQ-SEC2. We were talking about it because we've actually only had one patient in our Boston cohort with what we thought was a pathogenic variant in IQ-SEC2. I know you've had more in Australia, but the interesting thing is that we were having a conversation over the tea break about the fact that the Facebook group has grown now from, I guess, a couple dozen a year ago to now over 100 patients. And I think this goes back to what Ingrid was talking about in terms of the power of the community to provide the initial group of patients. It's, nobody's asking the parents to go get PhDs and become data scientists, but by organizing and by gathering, you're at least putting yourselves together, and it's the beginning of what could be a registry, and then you have somebody with an eye to the gene who can look at all the variants and say, yeah, these look like they're really the pathogenic variants. Maybe these couple reconsider and take that list to a big community like this, and if there's going to be a pan-Australia gene-agnostic natural history study, and which genes will you do first? I mean, my advice would be get as many people as you can together, because it would make sense to do the larger cohorts first just to get some traction and to get going. And so, so yes, there is some very, very specific research on IQ-SEC2 going on in Adelaide, and a very, very wonderful group of your colleagues whom I met the other day who are working on X-linked disorders, and really basic scientists who are thinking about the X chromosome and genes on the X chromosome, and there's some very funny things about how the X chromosomes turn themselves on and off that are being understood, and so there's a lot of basic work being done, and the nice thing is that group is very connected to Ingrid's group here, and you all meet regularly, and I've been able to be party to that the last couple of months, and I think there's tremendous potential to really do that translation that we talked about earlier.
I want to go to you, Steve, and ask you one question related to the kind of work that you're doing here at the Florey. One of the things that you've done differently from some other practitioners is that you've really kind of dabbled in several different models. So you've worked in some for-profit companies, you've done the clinical side and worked in a university setting. Now, as the director of the Florey, you did a big launch here with Praxis coming to town. I'm interested in your thoughts about how we do these things more quickly and maybe change the way we think about the best models for bringing drugs into development, for working out gene therapy. Are there lessons from this?
So I'm certainly not going to pretend I've got all the answers. All I've got is an idea of what I think should be done. And there are probably going to be numerous ways, but it's what became apparent to me was that there were many moving parts in getting an observation from a clinical diagnosis to a treatment, and most of us don't consider the entire food chain of that, from the physician to a lab, to a commercial entity, to a parent group, to a regulatory agency, to an investment company. And even drug companies themselves often pick up that process later in the stage when the science is sorted out, because they can't go back all the way. So it's important to get maybe a helicopter view and not be an expert in all of those, but get that helicopter view of all the steps, know who all the important players are in doing that, and then try to think about how do you establish the networks where you can connect that, and then sit back and watch, because no one person's going to be able to force that process, but you can certainly produce an environment and a mechanism where the right people come together, and that should then happen as a result of that interaction. And that's what I'm trying to think, and as a director of the Institute, thinking about who the different communities are. If we do something good at the Institute, it's great if we can obviously impact the careers of our scientists, because they're really important to us, but we also want to impact patients and families. We also want to impact society, and that we can impact society through patients, but we can also impact it by developing spin-out companies that come out of the Institute and then employ students from the university and other services, and then build health and wealth for the nation in other ways. And if you can make that work, I'm not saying we can, but if we can, and then that really sort of closes the loop. Because you see that really well in America. You see that, you look at Cambridge, Massachusetts, 20, 30 years ago, it was a parking lot, and now it is the highest concentration of biotech companies in the world, and it's not hurting them. It's not hurting MIT, it's not hurting anybody.
Well, and let me go to you, Anne, as somebody who is over there in Cambridge at Harvard. There are a couple of different sides to this, though, too. I mean, it's super exciting for families to see all this research, but also some of this stuff comes in, and the price, as we've heard today, can just absolutely knock your socks off. What are the challenges for medical professionals as you work with families? And, we're families. Lots of families don't have, tons and tons of money. I talk to people all the time, and they're making incredibly difficult choices and saying things like, we couldn't afford that for both our kids. We have 2 children with this disorder. How do we deal with these incredibly important questions of cost, of access, of being equal and fair?
I think this is why Dan's title this morning of Balancing the Hope and Reality is so poignant, because we all have a lot of hope and a lot of tremendous excitement for the science. And as scientists, there's a lot of new opportunity and technology that's within reach to find cures for one patient with Batten disease in my friend Tim Yu's lab, one group of patients with another disorder where there might be a handful of children helped, and yet the vast majority of patients we see face-to-face as clinicians are not even getting diagnosed with genetic disorders, even if we think that they're likely to be genetic, let alone having opportunities for specific gene-based treatment. And so there's this real tension between our excitement as scientists and then the reality of what we see for those of us who are also in clinics about the ability to provide access to the vast numbers of patients that we see. And one very tangible example was we were contacted as an institution to try to develop ASO therapy for a patient out of country with a very specific gene mutation, and it was actually a variant that one of Steve's papers had actually characterized, and so nobody was arguing that it was pathogenic. And at the same time, we had another baby in our own unit with the same gene involved, and the parents didn't have the resources to access the top-level scientists at the institution, just in terms of an access issue and the connections that they had, let alone the ability to start a foundation and those things. And so there we have 2 children, one abroad with resources, academic as much as financial, and a local family who didn't. And people come and say, well, what can we do? And shouldn't we start with the one that we can afford to do, because there may be some funds there, and then let everything sort of trickle down? And we said, we're a pediatric hospital, and we can't tell our local family, sorry, you're going to have to wait because we're going to take somebody from out of country because there's resources there. But that's easy to say, but then if you hold fast to those sorts of ideas, that it's possible that nothing will happen either. So I think we're really stuck. We're stuck because we were excited about the science. We want to move things forward. We don't have the resources to look at every gene. We don't have the resources to go deep on the science with every gene. And we certainly don't have the resources to make customized therapy for every patient. So I think our compromise has to be a little bit of this sort of hub and spoke model that Ingrid and I were talking about, where, we're not going to reinvent the wheel at Boston Children's Hospital and look at every variant at KCNT1 to see is it pathogenic or not, because Steve's looking at them already and publishing them, and so I think hopefully we can at least do some of what we hope to do by partnering across institutions. And so let's get the best of the basic science together with the drug development. To some extent, our institutions have to relax a little bit in terms of the conversations we can have with industries, which happens at MIT but does not happen at Boston Children's Hospital and many academic institutions for good reason, because we can't have inappropriate conflicts of interest. But I think there has to be a way to have those dialogues to push things forward. So it's a long way of saying that we have a lot of tension, and we can't have the answer be that we don't do anything because we can't do it perfectly.
Does anybody else want to weigh in on this? Because I can imagine it affecting everybody.
And if the question also relates to the high cost of some of these therapies, such as, I know there's a story happening in England now about a little boy, Harris Khan, who's trying to access Spinraza, and the UK regulators haven't decided whether or not that's going to be a reimbursed drug. I think it's just important that we continually challenge. I don't think we can sit from day one and say, yep, this is what's going to happen. But I think what we can do is that we continue with what we're doing, we engage. The price will eventually get set by a combination of the cost of developing the molecule, the appetite for the government and the insurances to reimburse, the pressure from patient groups. We know in Australia, Spinraza got reimbursed because of the SMA parent groups lobbying directly to the health minister. And that went from $600,000-700,000 a year to $30 a dose.
And by the way, you guys, that is why it is so important you're here. We cannot make changes if we don't come together at things like this.
Correct. So let's just keep challenging the system and let the government do their job. And until they're faced with that problem, they may not know how to solve it.
Anybody else?
I just had a little sort of counterpoint there that we have to really do it wisely. And I thought you were going to mention the other little boy in the UK. What was the name of the little boy that had the mitochondrial that Great Ormond Street got into terrible trouble with? Do you remember, Sophie? Oh, and the parents went to the legal courts about going to America for mitochondrial therapy. Charlie Gard, just come to my head. And there they really wanted very expensive treatment in the US. And it was not going to work. It was way too late and it wasn't going to work. So I guess we just need measure and we need wisdom. But it's so exciting when you hear about the drugs like alpha-synlipinase for Tom. I think that we have to be really good, and with the new senescence, so there's real hope, but we have to do it in a measured way.
I'm going to pause for a minute. Any questions here among our audience? I've taken some that we've gotten on our live feed. Does anybody here have a question? Yes, right here. I'll take you first.
Sorry, I just want to return the conversation to gene therapy because in the case of my daughter, she's got a recessive loss of function mutation, and my understanding is that gene therapy can probably hold the biggest promise for her. I have it on good authority that in a mouse model, knockout mouse model, seizures have been completely eliminated, all comorbidities ameliorated, and the life of the mice have been extended way past the usual lethality period. Now, this hasn't been published, but I know it's on the cards. What do I, as a parent, do to somehow find that solution for my daughter here in conjunction with the scientists that are working on it? How do I have the conversation with the medical community, maybe Professor Schaeffer, to actually look at that N of 1 treatment for my daughter? You know, I'm racing against time, and I just want to determine how big the mountain is, you know.
I think that you're right, that we probably do need to do some sort of gene therapy, but I was looking at Steve because, you know, we've had a lot of conversations this week about gene therapy and the issues along the way. So I think you're right. I think your job is to push for it to happen. But Steve, can you put some reality around that too, about what it is, what's involved?
From gene therapy in mouse to gene therapy in a person is a big leap. There's multiple, and Dan touched on this. He gave an example of how a gene could be delivered in the blood to get everywhere, but that's not always the case. And is there a human safe viral vector that you could put your gene in, that you can deliver, and how do you want that gene to go in every cell in the brain? Often these viruses aren't discriminatory. They will go in every neuron, in every glia. And so answering all those questions is just going to take time. So I think it's great a profile of a successful gene therapy project, you would want to see that it worked in the mouse. If it didn't work in the mouse, you'd want to say that's one of the steps along the way. I don't exactly know what how far along that project is, but there are numerous companies looking at developing gene therapy. My feeling is, and Dan gave the perfect example, cells that you can remove from the body, manipulate with a virus or another gene therapy method, and replace is going to be where the first things will happen. That'll happen with editing, etc. I think maybe in the brain, therapies that might benefit from a depot, where a small collection of cells can either release a drug or correcting a small nucleus, such as in something like a Parkinson's disease, might be where some of the first successes are seen. But therapies that require every excitatory neuron to be to have the right level of expression of certain gene might be a little bit further behind that. As each of these technical challenges is met, I think that we can start to ask more and more complex. And it goes back to that issue that Dan mentioned about the importance of basic science. Now we need, who would have thought that virology would have been important for CNS therapy 30 years ago, 40 years ago? And it's these unexpected collisions that occur in science. That all of a sudden, someone will make a virus that you can deliver to the blood and will get into the brain and will get in there, with high specificity. And that's the sort of thing that we're sort of waiting for, to have that moment. And then all of a sudden, you'll see this big explosion of activity.
So we don't have a good answer for you, is the bottom line. But the hope is, are these unpredictable advances that suddenly open up new doors. Everyone's heard about CRISPR. CRISPR's been in the news. CRISPR has probably accelerated our ability to do gene therapy by 100-fold. In other words, everything that's happening right now over in a year would have taken collectively many, many, many decades. It's been an astounding discovery. And so those are the type of advances that are going to sort of bring that distant future a lot closer. But it's just unpredictable.
And I guess I just wanted to add, was where I was talking about with Steve, was that once you've shown it works in the mouse acutely, say gene therapy, then you would need to look at chronic therapy. Is it safe 6 months out? Then you have to go to a higher primate model and test it there as well. So there are several steps before you would, with your hand on your heart, go to a child and use it in a child.
Yeah, I guess the only other thing I would add is that, I think with each disease and its known history and natural history, and when we're talking about taking basic science and preclinical studies into patients and children, the risk-benefit calculation is very much on the minds of the physicians, of the scientists, the ethics committees, and everybody. And so it's not a surprise that many of these novel therapies are seeing their first illustrations of success in cancer, because there you have a very, very known and often very dire prognosis, and we tolerate more risk. With some of the more progressive epilepsies, such as the CLNs, the Batten's diseases, and so on, there is a progressive history, well characterized by natural history, but we know that there's a progression, and doing nothing starts to become an active unacceptable, as opposed to sort of treading water. It becomes harder, and it becomes somewhat philosophical sometimes, but it's, is doing nothing acceptable when you've got an epileptic encephalopathy? We were sort of taught, well, if the seizures are gone, then the seizures are gone, and then you can stop. We all know that in many children, the seizures aren't the main issue. There are many other issues at play. But when you have a child who's stable, maybe who's walking, who's interacting, who doesn't have life-threatening convulsive seizures, how much risk are you going to take in terms of an invasive approach to therapy of an unknown efficacy? And we start to get to this tipping point where we know there's potential harm from some of these treatments now because we really haven't, we're just scratching the surface of delivery and how to do that safely, and we still have unknown benefit. And I think that's really hard because you're hearing about these little lights of success stories here and there for very sick children who really don't have a lot of good options where the outcome is dire. The risk that's tolerated in those scenarios is much higher than in kids who are stable, even if they're not stable and as well as they might be. If they're relatively stable, most people are not going to accept a very high-risk treatment.
I'm going to take one more question from up here. Yes, I think you had one. Who had the question? Yeah.
I did. I'm not sure if I should ask, but I just want to say thank you. This is just amazing, the whole GETA team putting this all together. The information is unheard of. You can't go to a doctor, a local doctor or a paediatrician anywhere in your state and hear this kind of information, so keep going. My question is around the genetic. I know we dabble a lot in all the different gene problems, SCN1A, SCN2A, and all the others, and that's amazing that they're all being evolved. But I just wondered, and you may already do this, but if you narrow down the actual gene problem, the actual coding problem, because obviously we all know with SCN1A, there's a whole spectrum of issues that come with it. You can be on one end and having 100 seizures a day, or you can be having 12 a week, and the comorbidities. When you talk about epilepsy, it's not just treating the seizures. There's a whole lot of comorbidities that go with that. It must be in relation to the makeup of that particular gene. I just wondered, is there anyone or any of you working absolutely together with one person's genetic problem, with their one little coding problem that is the problem, the issue, and all working together on that one? I just sometimes think if one person...
This leads you to Steve, for sure. This is your guy.
Works completely on that one person's issue and crack that, you might start to unfold the whole lot.
Thank you for that great question.
Anne touched on in their talk this idea of N=1 programs, and whether or not you can tailor a therapy that specifically addresses the genetic lesion in that one patient. That opens up, there's all the ethical and clinical questions, the issues that Anne raised, but so let's say we take Dravet syndrome as an example. If we were going to do that in order to test this proof of concept, as you're suggesting, I mean, we would have to believe that the variation in Dravet syndrome is driven by that differences in the genetic makeup. And often it's not. I mean, there are mutations that make the 1A gene disappear, and you truncate it here or you truncate it there. The difference may be due to the issues that Emma Palmer raised about genetic background. But if you were to fix the mutation in that patient, it would probably still correct those other disorders. It's just that by removing that one allele, you've moved it below the threshold. So we would have to be convinced that all these other factors, whether it's epigenetic, genetic background, and other things, needed to be corrected for that one person. And it's hard. I think it would be hard to embark on that because we don't even know what all those other factors are. We don't know what the myriad of other genetic background issues are. We don't know how epigenetics, how the biome might all interact to increase or decrease severity. What we could do is make a therapy that might help, and that's happening, that might help all people with truncating mutations in Dravet syndrome. And then from those experiments, and if we support that, from those experiments, you might see whether or not the people that are having 50 seizures a week and those that are having 2 seizures a week are all receiving similar benefit when you do fix the prime. That's my hope. That'll happen, and then we don't have to go down that way more complicated situation. I know what you're thinking, and we've had that similar thought. It seems like, I think, a very difficult problem, but then back to the N of 1, whether we want to fix a single patient, that is happening. It's happening now in the US. There are companies, Q-State Biosciences, that used to be Panomics, is embarking on that, and they're advertising that. There's the work happening in Anne's Institute. So we will get ideas around that. I don't think that'll ever impact more than those 1 or 2 patients because the FDA doesn't want drug development to occur and expand from a single patient and then blow out to a population because it circumvents established processes that control risk and all the other things that we need
And
to be
that's a very
careful of.
important thing. Let me pause for a sec, because we really are almost out of time. Did you have a comment? Yes.
I was going to say that related to that point, though, there are many instances where specific mutations have actually told us a lot about the condition and the underlying pathology and pathomechanism. So, for example, Steve's work in SCN2A, there's a couple of recurrent variants, one of which, and each presents clinically quite differently. And Steve has shown that one causes a gain of function and one causes a loss of function, which then leads to the idea that they will probably need different treatments. Understanding that about those two specific mutations has then allowed that to be extrapolated to children with other mutations who present like with these clinical symptoms or these clinical symptoms, and that can expand to treat a lot more children with the condition. So we can learn a lot from individual mutations that may end up for treatments for more children.
And Emma has a thought as well, yes?
Just a really quick thought, because when I hear your question, of course, if that was my child, I would want the gene test to come with, and this is the drug, and this is when you start. That would be absolutely what I would want. Can I try and just put some hope back here that what we're saying about a pan-Australian international network of knowing which children around the world have which genetic changes in which genes and how that's affecting them, that it has to happen in order for drug companies to invest, to say, there's a groundswell, there's enough children here, or also, as just to come back to what Catherine said, that someone has done the work in a family in France, and that's translational to a family in Australia. So it may not sound sexy, but why we're trying to come back to that story is to lay those groundworks for what you want and what you deserve to try and give it the best chance to work.
Yes, of course.
Just to follow up on what Steve said to you about SCN1A, I think it's really important. 40% of the patients have truncation mutations. The one treatment might work for all of them. So you don't need it personalised to your mutation, because it sounds like that's what everyone thinks we need. And
That's an incredibly important point.
So it may be that one treatment that can up the normal allele, the normal copy of 1A, to replace, if you like, the one that's not being made properly might be enough to work, and that's what people are looking at. So that's one thing. The second comment I wanted to make, which is slightly also goes on to what Steve was saying about your genetic background and how with a good genetic background you might be much better in terms of cognition than someone without a good genetic background. It's not anything we have control over. We have 19,000 genes and they've all got variants. But we're just starting a study with, I think, 6 European centres to do genome sequencing of 1,000 people with Dravet and to try and start teasing out why one person with Dravet has severe intellectual disability and another one doesn't, has mild. And so if we haven't got your DNA, please send us your DNA.
You know what, thank you for saying that as well, because I just don't think I can say this strongly enough. I've heard a lot of people say, oh, I'd like to be involved, I'd like to help. We have room for you in our hearts, in our group, with the science. So reach out to Ingrid if you're interested in the natural history study, or if you're part of, Dravet and you want to have that be part of this. And connect to Danielle and Chris and me. Give us your names, give us your contact details, tell us what you like to do. How can we put you to work? We've got plenty, and we can be stronger that way. Kelly, I saw that you had a question, and then we're going to have to wrap it because Ingrid does have to make that flight.
I just wanted to say, I remember about 15 years ago, I put the question to Ingrid as when are you going to stop looking at medications to stop the epilepsy and start looking at fixing the gene mutation itself? And to hear the last 5 years especially has come such a long way. There is so much hope there, guys. This work that these guys have done in such a really short time is massive, and our kids are being better treated now than they were 10 years ago. And their quality of life is probably better than they would have been 15 years ago. So in the last 5 years especially, the work that these guys have done for the kids and for your families has been, even though it sounds like a long time, 15 years, it's not. It goes really fast, and it's great to see that work is being done to stop the mutation altogether.
I think that's a brilliant point.